Hello, Namaste. Welcome back to online classes brought to you by Vivekananda PU College. I am Supreeth Casey. In today's class, we are going to discuss whatever the chapter left behind, the concept left behind in meiosis, that is meiosis 2. Okay. So we are talking about the chapter called as a cell cycle and division, wherein we have studied mitosis, we have studied meiosis. Meiosis is nothing but it is a process in which the germ cell, when it undergoes cell division, and it will form a daughter cell with a reduced chromosome number by halves. If you have a chromosome number that is 46, and at the end of the cell cycle you will be having a 23. Okay. The concept is like this. See, I have a parent cell which is having a chromosome number of 2n, okay, and it will form two daughter cell, okay, by the process called as meiosis 1. Meiosis 1, okay. After meiosis 1, it will enter to the stage called as meiosis 2. Meiosis 2, and again you will be having a cell that is of 4 daughter cell with a chromosome number n, daughter cell. See, look at the daughter cell. There are 4 daughter cell with a chromosome number n, or it is known to be the haploid number of chromosome. This is how the meiosis is happening. And in previous class we learnt about what is meiosis, and the whole set of meiosis is divided into two, that is meiosis 1, meiosis 2. In meiosis 1 again we will be having 4 stages, that is prophase 1, metaphase 1, anaphase 1, telophase 1. In the prophase 1 we will be again witnessing 5 more stages: leptotin, zygotin, pachytin, diplotin, as well as diakinesis. Now we will be moving to the second step, that is meiosis 2. This meiosis 2 is which is very simple, and it is having a similarity with the mitosis, because you don't have a much difference here. In meiosis 1 you had a recombination, you have a synapsis, okay, you have a crossing over. But in meiosis 2, nothing like that. How the mitosis is happening, the same order will be followed by meiosis 2. After as you know, first will be karyokinesis, the nucleus will divide, and after all the process, when you reach telophase 1, you will be finished with the distribution of the cellular components, cytoplasmic components. Now distribution of cell components. Now the cytoplasmic component also will be start to dividing, called as a karyokinesis. After this karyokinesis, you will be having a small gap called as interphase, a gap or a state in which the cell is taking a little rest in between meiosis 1, meiosis 2. The state of rest may be different in different organisms. Okay. Now, in meiosis 2, there are 4 major steps. There are 4 major stages. What are those stages? Okay. In the case of meiosis 2, you can have 4 main stages. One is called as prophase 2. Prophase 2. Next one is metaphase 2. Prophase 2, metaphase 2, then anaphase 2, telophase 2. Okay, anaphase 2, telophase 2. Telophase 2. Okay. So this is about the different steps under meiosis. Prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay. What exactly mean by prophase 2? What exactly mean by metaphase 2? What exactly mean by telophase 2? What are the steps that we are going to study under this? We will talk about that. Okay, again I have a chart. Okay, fine. Let us see that. Yes, I have a chart to discuss. Look at the chart now. Yes, this is a chart which shows the cell division. Carefully observe the chart for a minute. Okay. Now let us begin the concept. Fine. As you can see now, the interphase or interkinesis is a state in between meiosis 1 and meiosis 2. Say I have a meiosis 1 here and I have a meiosis 2. In between meiosis 1 and meiosis 2, I have a state, a period of a small rest, or the period of a preparation of the components, called as interkinesis. Interkinesis. Okay. In interkinesis, again the cell is very normal. The cellular components are behaving very normal, as well as you can find out the chromatin fibers, which is loosely aggregated. Okay. And you have a cellular envelope which is protecting your nucleus, nuclear envelope which is protecting your nucleus, and all the components are doing a perfect function. Yes. Now this structure, if we need to enter the stage of meiosis 2, what will happen? Again, same as of mitosis, the nuclear envelope, okay, nuclear envelope start to disintegrate. When the nuclear envelope is start to disintegrate, then whatever the content present inside the nucleus, okay, which will move forward for the other step, the chromatin fiber will be undergoing a condensation. Okay, condensation, chromatin fibers undergoing condensation. Okay, the condensation is occurring and you can witness the chromatids. When the chromatin fiber is undergoing a condensation, it will form a chromatid. Okay, chromatid. Then we already know, after the process of telophase 1, you have a 23 pair of a chromosome with a copy. Now only thing which matters here, the distribution of a chromosome with respect to the cell. Now when the interkinesis is over, in second prophase, the cellular envelope, nuclear envelope start to disintegrate and chromatin fibers will be undergoing a process of condensation. In that case, the aster will start to move to the particular, look at this, to the pole. This is a pole, this is a pole, this is a pole, this is a pole, and this structure is stabilized by the presence of microtubules. Look at here, microtubules called as spindle fibers. There will be a presence of spindle fibers there. Now the whole set of a chromosome, which becoming a very tight or condensed manner, they will be attaining in the equatorial position, at the center equatorial position, and that is held by your microtubules. Now this is called as a metaphase plate, and you will be witnessing second metaphase in meiosis 2. This is about metaphase plate. Okay. After this metaphase plate, you have a centromere that is again undergoing a division, and the whole set of a chromosome after division, each centromere will be pulled apart to the poles, successive poles. See, pulled apart to the successive poles. The shape will be like scissor. Okay, the X shape will be attained by the chromosome in the phase of anaphase, and it will undergoing a division like this. Now the structure will be moved towards the pole. This is the cell. The whole structure will be moved towards the pole, move towards the pole, move towards the pole. Okay, this is what happening. When they reach to particular pole, okay, reach to the particular pole, and the force exerted by your aster, okay, they become weakened. Even the spindle fiber force which is helping to pull in the chromosomal content will be becoming little weaker. Okay, that is the ending of second anaphase. Now, after attaining to the particular pole by the chromosome, now the structure is ready for the telophase, where a slowly nuclear membrane is start to form. Okay, simultaneously whatever the cellular component become hidden, they start to visible. Even the condensed chromosome, okay, they become decondensed, loosely aggregated. Now the cell is normal. So at the end you will be witnessing 4 cells. You will be witnessing 4 cells. Okay, this is about the telophase second in meiosis 2. This is very similar to mitosis. So the second meiosis is always termed as a mitosis also. Okay, this is about meiosis in meiosis, that is second phase of meiosis in the whole set of meiosis. So we have studied meiosis 1, meiosis 2, they are the part of meiosis a whole, and in meiosis 1 we will be having prophase 1, metaphase 1, anaphase 1, telophase 1. In again meiosis 2 you will be having prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, this is a ending story of meiosis. Let us study what is the significance of your meiosis. Why meiosis is so important? What is the significance of meiosis? Why meiosis is very important, that you must know. What are the significance of meiosis? So they say significance. Significance of meiosis. In meiosis, it conserves chromosome number in a species. It conserves chromosome number in a species. It conserves chromosome number in species. The chromosome number of a particular organism is conserved by the meiosis, the process of meiosis. Okay. Then recombination is possible by meiosis. Recombination is possible by meiosis. This recombination even leads to evolution. Small changes in the organism according to their habitat, according to their adoption, according to their parents' character, all will be possible by the meiosis, which is leading to the evolution. This is the significance of meiosis. Now let us study why this, what is the difference between the mitosis as well as meiosis. Difference between mitosis as well as meiosis. This recombination will be leading to variation. Recombination is lead to variation. I just forgot to say this. This recombination lead to variation, that is happened by your meiosis. What is the difference between mitosis as well as meiosis. Let us study difference between mitosis and meiosis. Mitosis, meiosis. What is the difference between mitosis and meiosis? Mitosis can be, it is a very important process, as well as you can witness mitosis in somatic cells. Somatic cells are the cells where growth and development is happening, the cell which is responsible for the growth and development. This meiosis is happening in reproductive cells. Reproductive, the cells which are responsible to produce sperms as well as ovum, egg. Okay. So in mitosis, it is happening in somatic cells. Meiosis, it is happening in reproductive cell. At the end of the cell cycle in mitosis, 2 daughter cells are formed. Okay. But at the end of meiosis, 4 daughter cells are formed. In the case of mitosis, diploid number of a parent cell, okay, is undergone a process, and at the end of the mitosis, the parent cell, the daughter cell also having a 2n number of a chromosome, nothing but diploid character is maintained. But in the case of meiosis, diploid condition is converted into haploid condition, where the daughter cell will be having haploid number of a chromosome. If it is 46, they become 23. Here 46 will be conserved. In the case of mitosis, we cannot find crossing over. Crossing over is absent. Here crossing over is present. Crossing over absent, crossing over present. In the case of mitosis, recombination, we can't see the recombination, but in meiosis, recombination present. Recombination is present. Okay. Then in the case of mitosis, synaptinimal complex absent, but in meiosis, synaptinimal complex is present. These are the some difference of mitosis and meiosis. Okay. So I hope you have understood this class of meiosis 2. Okay. I think you better to watch the class together of meiosis 1 as well as meiosis 2 all together. Okay. It will become more help you to more understand the concept. So I hope you have understood this concept, and this is the time to wind up the class. So thank you so much for your patient listening, and read this concept well, the significance of mitosis, significance of meiosis, their difference. This is very important, and this is the ending of a chapter called as cell cycle and cell division. I hope you have liked it, and please read this concept once again in your textbook. Thank you so much for your patient listening. Have a nice day. Take care, stay at home, stay safe, and read well. Thank you so much.